Thank you guys so much. Just like Kate said, it's my first time being at an in-person conference with DSF. I'm so excited to be here, to meet families, to finally meet so many people that I've only seen in a Zoom box for the last 2 years, actually in person. So I do have my PhD in biomedical research. I was trained as an academic scientist, but I've also been a caregiver to a child with a really severe neurodevelopmental disorder. And so this job is incredibly meaningful to me because it's bringing together my experience as a caregiver, and so many of your stories resonate with my own, and getting to use that experience for my personal life, honor the life of my son through that, as well as use my scientific training to hopefully help families that are in a similar position to I was. This job's incredibly meaningful, and so thank you so much for letting me be here. I'm just going to kick the conference off with a scientific primer. We're a little late on getting started, so I'm going to try to speed my talk up just a little bit so we can get back on track. So what I'm going to cover is really just how research has progressed for Dravet syndrome, from the beginning of it being described and then all the way up to DSF being founded, and the changes that have really improved, hopefully, patient care since then. And I also want to focus on the role that patient families have had in this. And then I will end just giving families a few tips about the conference, a few things about what to expect, and hopefully that will get us kicked off to a really great time. So starting out with a little bit of history, Dravet syndrome was first described in 1978 by a French neurologist, Dr. Charlotte Dravet, and at the time it was called severe myoclonic epilepsy of infancy, or SMEI, and you may even see that nomenclature still being used occasionally today. But in 1989, they renamed it to Dravet syndrome because they were really worried that with infancy in the name, there was a lack of recognition that this is a lifetime disorder that affects patients, not just in infancy. And so, at the time, we still didn't know what caused it, and in the early 2000s, there was a big revolution in our understanding of genetics. And in 2001, that led to researchers finding this link between Dravet syndrome and mutations in a gene called SCN1A. And everyone thought, okay, now we know the cause, things are going to change. And almost a decade later, families were feeling frustrated because research had progressed some, but there weren't impactful changes happening to clinical care. And so that's what led to the formation of the Dravet Syndrome Foundation in 2009. And DSF operates under the mission to aggressively raise funds for Dravet syndrome and related epilepsies, to support and fund research, to increase awareness, and to provide support to affected individuals and their families. And I think just as a quick snapshot, you can see what an impact has been made when you see the amount of dollars that DSF has directed towards research since they funded their first grant in 2010, $5.6 million. And in addition to just that monetary, we can put numbers on it all day, but something else that's less easy to quantify is how DSF has acted as a convener for all the stakeholders in this space. And when you look around this room and you think about everyone Kate was just introducing and talking about being here, I think that exemplifies that. DSF is the central hub that brings together patients and families. It brings together scientists, clinicians, and clinician researchers, industry members, government regulators, everyone with a vested interest in seeing outcomes improved for these patients. And so what has that done to the field? If we fast forward to 2022, where we are now, some big things have happened. There are 3 medications with FDA-approved indication for Dravet syndrome that have received that approval just since 2018. There right now are at least 5 interventional clinical trials that are currently enrolling in the US, and there are more planning to start soon. Disease-modifying therapies are on the horizon, and I think that holds a lot of potential for really revolutionizing the way these patients are cared for. research is really booming. If you look at just this simple graphic, looking at PubMed, which is where you can find a directory of papers relevant to human disease, and if we search for either the gene name SCN1A or for Dravet syndrome, you can see that in 2001, when the gene was identified to be associated with Dravet syndrome, there were less than 20 papers a year coming out at that time. And if you look over the years, you can see in 2009, when DSF was founded, that those numbers had begun to climb, but they were still low. And now, as of 2021, there were over 250 papers published, just listed on PubMed. And in addition to that increase in publications representing the increase in research, we can also look at NIH funding, which has greatly increased over those years. So in, I believe, 2008, NIH, which is the biggest governmental funding of health-related research, they had funded about $2.6 million that year towards research. Since 2014, so about 5 years after DSF was founded and began really funding these seed grants, every year since 2014, the NIH has directed over $10 million a year towards Dravet syndrome, and there's an ever-increasing awareness that this is important, and we need to fund even more of this type of research. So I want to just give a few examples of things that have happened, discoveries that have happened that I think have really impacted this picture. And so the association with the SCN1A gene, I think, really opened the door to enable discovery. And the SCN1A gene codes for a sodium channel called NAV1.1. And every single cell in your body contains DNA. Contains the full DNA blueprint for all of your genes for your body to function. And those genes get regulated in different ways so that you can have a variety of cell types and tissue types and organs all doing unique functions. And so now that we knew the gene that was predominantly impacted in Dravet syndrome, scientists could begin to understand where is that gene expressed and what does it do and why would that cause this syndrome. And so scientists were able to identify that this sodium channel made by SCN1A is primarily used by a subset of cells in the brain called inhibitory interneurons. Now, like I said, everyone has 2 copies, and what they found from looking at samples from patients is that most patients were carrying a mutation in 1 copy, and that led to a reduction in the amount of sodium channels that they could make. And so now these inhibitory neurons that are supposed to have these sodium channels actively bringing sodium ions in and out of the cell and allowing neurons to send electrical communications for proper brain and body function, suddenly they only have half as many of those sodium channels. And suddenly they can't function as well. This type of genetic condition is called a haploinsufficiency, and it just means you have about 50% less gene function. And why this was such a big deal in these inhibitory interneurons is because these neurons are supposed to kind of be the brakes in your brain. So when cells are talking and you have a lot of electrical activity happening, these cells are supposed to say, okay, calm down now. And when they're only working at part of their function, they can no longer slow that down. And so that's kind of the current hypothesis about why this mutation leads to Dravet syndrome and to seizures. So that was one way that understanding the gene led to discovery and understanding of the syndrome. Understanding the gene also allowed researchers to make a whole new toolkit to study the syndrome. And these toolkits are a lot of the ways that they were able to discover more about the cause. And so at the conference this weekend, you're going to hear about a lot of different types of models, mouse models, where they're able to use the incredible genetic tools that we have to mimic mutations that may cause Dravet syndrome in patients in a mouse. Because even though a mouse is very different than us, a lot of the genetics are very, very similar. And we now have several mouse models that can show different or have different mutations in the SCN1A gene that are relevant to human mutations that we can now study more about Dravet syndrome in. And you're going to hear about some of these tools that allow researchers to manipulate the genetics in the entire animal, in a specific subset of cells, or even the ability to turn the mutation on and off in the animal. And while that's not going to be possible in a human in the same way, it allows us to ask some of these important questions that are relevant to human disease. There are also, you're going to hear about zebrafish models. These also were able to interrupt the SCN1A gene in a zebrafish, and then scientists can study epilepsy phenotypes in a fish. And that seems kind of crazy. A fish, how is that going to mimic a human? But what's really amazing is that these fish seem to have a really similar response to anti-epileptic drugs as human patients do. And so you can do really big drug screens in these fish because they're small and they develop fast, and it can lead to really important medical discoveries for new drugs. We also have cell models that can either these animal models or from patient cells. So we can take blood or skin samples, turn those into stem cells, reprogram those stem cells into neurons, and now you can study a patient's cells in a dish. All of these tools have allowed us to discover incredible things and have led to a lot of the treatments that you're going to hear about that have been developed or in clinical trials or are approved already for patients. In addition to these basic science impacts, we also know that there have been changes to clinical care that have come over the years. Diagnosis has come a long way. The association with a specific gene has allowed for genetic testing of patients that can help either guide or confirm diagnosis. It's important to note that this is still a clinical diagnosis, which means a doctor is making the diagnosis based on signs and symptoms in the patient. You're not required to have a genetic mutation to confirm the diagnosis, but it can help doctors feel more certain that they've made an accurate diagnosis, and it can certainly guide treatment. Genetic testing has continued over the years to become more and more accessible, and studies have shown that diagnoses appear to possibly be happening much earlier now that we have more access to genetic testing. And in addition to just the access, I think awareness of Dravet syndrome has increased exponentially. More doctors know about it, more people know about it, and the importance of genetic testing has also increased. And so even if a doctor doesn't know Dravet syndrome, they may be more likely, when they don't have an answer in a young child with epilepsy, to order a genetic testing panel when they get the SCN1A mutation back that can help guide them towards a diagnosis of Dravet syndrome. It's important to note that there are probably still many adults that still have never received a diagnosis because they were young before the advent of genetic testing. And so there is still work to do. And not every patient with Dravet syndrome has an identified gene mutation. And some of those that do don't have an identified mutation in the SCN1A gene. So there are a few other genes that can contribute to Dravet syndrome, and there are some patients that have clinical Dravet syndrome but have never had positive genetic testing. So while we've come a long way, there's still work to do in understanding more about the genetics. In addition to that, we have seen clinical studies multiply. Back in 2009, when DSF was founded, there were no drugs approved with an indication specifically for Dravet syndrome, and now we have 3. Studies have also delved deeper into understanding the symptoms, the comorbidities, such as gait and motor disruptions, sleep, seizure types and triggers, cardiac function, and SUDEP, just to name a few. Studies have helped uncover the incidence of Dravet syndrome. A study funded by DSF in 2015 was able to estimate that this rare disease occurs in about 1 in every 16,000 live births. And in addition to that, we have seen an ever-increasing amount of inclusion of the patient and caregiver voice. And that's just so important. We need to hear what's important to families, and that should be helping to guide the research directions. Several studies now have specifically highlighted the totality of impacts of Dravet syndrome directly on patients and caregivers and their families. One example is a study led by DSF in 2017 that specifically highlighted caregiver concerns across the spectrum of medical and quality of life issues. And these are all published in scientific journals, and so they're available to families, but also to clinicians and scientists and researchers that can help guide their research programs based on the most pressing needs for families. In addition, just this last February, DSF helped to hold a patient-focused drug development meeting. This was an opportunity for families to engage with the FDA, as well as other important stakeholders in industry and academia about what life really looks like day to day with Dravet syndrome and what those most important needs are. You can watch the recording of that meeting at dravet-el-pfdd.org, and we now have a report from that meeting called the Voice of the Patient Report, where we have incorporated all of the findings from that day, all of the learnings, into one report. It's shared publicly. Anyone can access it now, but it will also be shared with the FDA. And now when the FDA is making decisions about drug approvals and clinical trials for Dravet syndrome, they're really able to take the patient voice into account by pulling out this Voice of the Patient report and making sure when they're weighing benefits and risks that they're weighing what's most important to families. You know, all of this has been incredible progress, and I've only been able to touch on a few items of progress. But I don't want to just say, pat on the back, it's all done, because we know there's still so much more to do. Seizures and comorbidities remain so burdensome for families. A lot of those studies that I was mentioning that are looking at comorbidities have mostly been trying to describe and understand what it looks like in a patient, and there's very little guidance on treatment for many of those still. Mortality is still high. SUDEP is still poorly understood. Supports for adults are particularly lacking. Transition of care is difficult, and everything about becoming an adult with Dravet syndrome seems to be a challenge for families as they feel like they have to recreate the wheel of everything they built for the last 18 to 20 years as they move into adult system and services. Disease-modifying treatments are on the horizon. There's currently a clinical trial for an ASO. There are several other genetic therapies that are hopefully going to reach clinical trials very soon, but they're not here yet. Clinical trials don't provide access to every patient as they're trying to determine if this treatment is going to be effective. And so there's still so much work that we need to do to get there. So what can you do today? You can get involved, you can get informed, and you can get going. You've done the first step just by being here today. But you should really continue to stay connected to DSF. We regularly make sure and to share opportunities to raise money, to help support research, and to participate in research efforts yourself. Coming to this meeting, watching our webinars, coming to our online conferences can help you learn about research that's ongoing so that you're the most informed and know what opportunities are available for your family. And when a study does seem like a good fit, you can discuss participation with your healthcare providers. And it's not always participating in a major clinical trial. There are regularly survey opportunities. That's a low-risk way that you can contribute your voice to research. You'll find a few examples of surveys that you can participate in your registration packets. But if you want to know about more opportunities, you can always reach out to me. And I just want to remind you all that family-led fundraising is the main way that DSF has been able to fund $5.6 million of research. Every single effort helps. Nothing is too small, so I encourage you to get involved. All right, and so then I'm just going to end with just a few tips for the conference. I want to encourage everyone to ask questions. That's my biggest advice. No question is too small or too silly to ask. I can't tell you how many times I've been too scared to ask a question in a talk and then kicked myself when someone who I thought was much smarter than me asked the same question. So just find some bravery and ask it. Take notes on things you may want to look up later, but remember, most of these sessions are going to be recorded, and so if you miss something, that's okay. You can go back and rewatch. And for families, a lot of these talks are going to feel heavy. Not only is this really dense information, but it can be overwhelming to hear this many talks back to back about all of the different comorbidities and struggles that you live day to day. It is okay to take a break. It's okay to take care of yourself. Like I said, these will be recorded. You can come back later and rewatch them. So make sure you're taking care of yourself this weekend too. Right after my talk, we're going to have several industry talks. So I just wanted to give you a few tips on interacting with companies, because sometimes that can feel scary. You want to ask questions, but you don't really know what to ask. So a few of my tips are: avoid sharing with a company if you've been involved as part of one of their ongoing clinical trials. They try to stay, ethically removed from the patients. They're not always involved. They're not involved in picking the patients that enroll. So try to refrain from using those specific examples. Avoid sharing adverse treatment-related events or seeking direct medical advice from these companies. Those are questions to take to your healthcare provider. But what you can ask about is what treatments they may be developing, what trials they might have going, what treatment they're currently marketing that might be helpful to a patient with Dravet syndrome. You can ask about current informational materials that they have or resources, but you can also tell them, hey, this is a resource that would be really helpful to me, or this is a way I would like to see information. And lastly, there's other ways often to engage with some of these companies other than just to participate in a trial. So you can ask about that too. I think that is it for my talk today. If you have more questions, you can always email me, veronica@dravetfoundation.org. If anyone has a question right now, feel free to ask. Other than that, we'll move on into our industry sessions. So thank you, everyone.